This is my 1.2 Task 2 assessment on the process of muscle contraction. I'm going to attempt to explain the process of an individual responding to a sporting environmental stimulus and causing muscular contraction to perform a sport-related skill. We have a nerve transmission, the neuromuscular junction, and sliding filament theory. The stimulus in the sporting environment starts the process and is acknowledged by sensory organs. In this case, a football is hit as a shot towards the goal, and the goalkeeper sees this. The information seen by the eyes is processed and is sent throughout the body as an impulse. This impulse is sent to the brain through the relay and the motor neuron. Once that impulse reaches the brain, a decision is made to contract the muscles so that the body can block the ball before it goes in the goal. The impulse leaves the brain, travelling down nerves until it reaches a muscle. When it reaches the muscle, it arrives at a synapse called the neuromuscular junction. Acetylcholine, or ACH, is released into the synapse, and this allows the impulse to diffuse across as it depolarizes the motor end plate. The impulse travels from the junction to the muscle through the T-tubules. This impulse causes calcium ions to be released from the sarcoplasmic reticulum. At rest, the myosin arms are energized by a hydrolyzed adenosine triphosphate in the form of adenosine diphosphate and an inorganic phosphorus. The breakdown or the hydrolyzation of the ATP gives the myosin arm this energy. The troponin and tropomyosin also cover the actin binding sites, where the myosin attaches to form a crossbridge, so this cannot take place. If allowed, the myosin arm would pull the actin attached to the walls of the sarcomere, called the Z-line, into the center of the sarcomere, regarded as the H-zone. The A-band of the sarcomeres is the section of the sarcomere where the myosin arms are, and the I-band is the point starting in one sarcomere where the myosin arm ends, and ends where the myosin starts in the next sarcomere. It will slide together to shorten the muscle. Calcium ions during rest are stored in the sarcoplasmic reticulum, but once an impulse has been diffused into the muscle fiber, the calcium ions are released into the muscle fiber as well. The sarcoplasmic reticulum is wrapped around each myofibril in the muscle, so the calcium can aid the myosin to form cross bridges and perform contraction. As the calcium is released, it bonds to the troponin that allows the displacement of both troponin and tropomyosin. Once they are displaced, the cross bridge can form. Diphosphate and the inorganic phosphorus make the myosin arm attach to the actin binding site and is used to make the arm pull the actin across the center of the sarcomere in a sliding motion. The actin slides into the H-zone, making the sarcomere shorter. To break the cross bridge, there has to be another adenosine triphosphate, or ATP, ready to be used. The energy is actually used to break the bond. The new ATP is broken down into ADP and an inorganic phosphorus as soon as the bond is broken. This is then used to create the bond again, and therefore the movement is repeated. During sporting activities, muscles have to contract and relax in different ways. The four ways are concentric, eccentric, isometric, and isokinetic. Concentric contraction shortens the muscle during the actual contraction. A sporting example of this would be the lifting face of a bicep curl. The next type of contraction is eccentric contraction. This is where the muscle lengthens during the contraction. A sporting example of this would be the lowering phase of a bicep curl. The third of the four types of contraction is isometric contraction. During this contraction, there is tension created, but the muscles do not change in length. A sporting example of this would be the ski sit. The last type of contraction is isokinetic. An isokinetic contraction is where the muscle contracts and shortens at a constant speed. An example of this would be the isokinetic dynamometer.